Friends, welcome back in another video lecture from Microbiology with Shagnik. This is the continuation of our previous video of ELISA lecture series, where we have talked about the basic introduction of ELISA, what enzymes that we need, and the substrate that we have to add in along with the enzymes, okay, and the definition of ELISA. So today we are going to talk about what the simplest form of ELISA is, okay. So let's talk about that. So let's take a hypothetical example. Suppose this is a person who have a infection. So in the laboratory you need to determine what kind of infection is. So you inject here the serum from that particular patient, okay. Now this serum must contain the antigen, okay, that you are suspecting, right? So you add the serum into a container containing microtiter well, okay. So what you can say, you can you add the serum into the microtiter wells, okay. So if the antigen that you are suspecting for are present, then they will going to be attached, or you can say adsorbed onto the surface of the microtiter well, okay. After that you will wash it, okay. That means the antigens that are not that are unbound, suppose they are present here, they will be removed. You will only have the bound antigens, okay. Now you will go add a antibody that is complementary, okay. Now I think it is visible, complementary to the antigen of interest, right? Okay. So suppose if the patient contains the antigen that you are looking for, then the antibody will easily bind with the antigen of interest in the present in the walls of the wells of the microtiter plate. This type of containers are known as microtiter well, okay. Okay, so they will bind with them, okay, and this type of antibody is also attached with a enzyme in its Fc portion, okay. So this portion of the antibody is referred as Fc, and this portion in which it is interacting with the antigen of interest is Fab, okay. This is fragment of crystallizable, this is fragment of antigen binding, okay. So so you have an enzyme linked antibody here. Now when you are going to add a colorless substrate, this colorless substrate in the presence of enzyme will give colored product, and the intensity of the colored product here will tell us that yes, the antigen that we are looking for are present here, okay. And you can also determine the amount of antigen present with the help of various colorimetric measurements, okay. So that is all about the basic ELISA technique, that is direct ELISA technique. But there are some problems here, there are some disadvantages. That is the reason why scientists developed this technique further, okay. So what are those disadvantages? Disadvantages is, suppose when we are talking about a very complex samples containing various mixtures of antigen, okay. So along with the antigen of interest in the microtiter well, there will be also another antigen that are not antigen of interest, okay. So the amount of antigen interest is very low, so there will be a chance that it will reduces the sensitivity, okay. As the amount of antigen of interest is very low, so low amount of enzyme linked antibody will bind over here, and substrate after upon adding on substrate, a very small intense light will come from them, okay. So the sensitivity of the result will be very low. That is one point. And second point is, when you are conjugating a enzyme with antibody, the affinity of the antibody towards the antigen will be reduced. Again, when you are conjugating a enzyme with the antibody, the affinity of that antibody towards the antigen Here I have used a wrong term. It should be specific to the antigen. Okay, not complementary. Sorry for that. So where we are, so if you use a antibody that is conjugated with the enzyme, then the affinity of that antibody for the particular antigen will be reduced. Okay, this will in turn reduces the sensitivity. These are the two major disadvantage of direct ELISA that you need to know, and that are very important, because on the basis to resolve those problems, scientist has developed the further techniques that are indirect ELISA, that we are going to talk about in our next video, that provide a more intense color. So the in case of complex mixture that I have mentioned previously, the problem that was occurring in case of direct ELISA will be resolved in indirect ELISA. And there will be another technique that we are going to talk about is sandwich ELISA, that will resolve the problem of sensitivity, that when we are going to talk about that sensitivity, because here we are only countering the antigen via one epitope, right? And in case of the sandwich ELISA, we are going to counter the antigen via two epitope. That also increases the specificity of our result. Okay, so that's it for today. I hope this video will be helpful to you, and if you like the video, so please like the button, share with your friends, and subscribe to my channel. Thank you.